just to overview some of the things we've covered. So we've shown how you can use the gene function discovery pipelines in KBase to identify candidate genes for functions of interest. Candidates could be proposed from gap filling or pathway predictions, annotation apps, alignments, blasts, and many other algorithms. And you can pool now the closest experimental structures associated with these genes from PDB using two different apps: our genome-wide query app, which gives you a wide but shallow pool from PDB, and our query app, which gives us our gene-specific query app, which gives us a deep query from PDB with more hits. You can export gene candidates as FASTA, generate structures in Google Colab. Again, if you have questions, like QZ is the one that did this, so she can, if you have questions on how to do that, she can give you some information. Then you can import those computational structures back into KBase or query and analyze those structures directly in PDB. And I'm going to show you something in a minute that's kind of connected to why we have these in both places. But you can use PDB query and import apps to pull structures with desired functions or substrates co-crystallize into KBase. You can query based off of UniProt or sequence or compound structure for the ligand that's co-crystallized. So you can query and EC numbers. You can very rich query, and just encourage you to look at that app. And in particular, look at the PDB query, because the tools are very, very good in the PDB site itself, and quite capable of doing a lot. And we only offer a subset at the moment in KBase, but I hope to keep building that out. You can align computational experimental structures and in the PDB site. So and this is the big thing. Like, the thing I envision is you'll find structures of interest, you'll get the sequences, you'll fold them, you'll put them in KBase just for documentation, but then you take them to the PDB site and you do these alignments. And then the idea is you would screenshot these and then put these in your KBase narrative to help document the science you're trying to do. So the KBase is, in a lot of ways, it's an analysis pipeline, it's a data compilation and sharing pipeline, but it's also a publication pipeline. Like, the narratives are designed to be a tool to share your ideas with the research community. And that's a way to do that. Otherwise, it's hard to make these stories and have the data there and every step that you did captured in the story. That's why we call them narratives. So comparing PDB complexes and co-crystallized ligands with complexes in reaction substrates and models. I'll get to this and more in a moment. I'll actually demonstrate this. So the idea is, it's one of the great values of PDB, particularly the experimental structures, as Stephen and others have mentioned today, is the co-crystallization information. It provides a wonderful context on the models that you build in KBase, which are based often on homology. But so that PDB is like an independent source of experimental validation for annotations that you make in KBase. And what's nice is, even when our leap to PDB is based off of homology, what AlphaFold now lets you do is go a step farther. You can generate an AlphaFold structure for your sequences in KBase, many of which will be totally new and never seen before, because they're from an isolate, or they're from an environment. And then not only see if that sequence is homologous by sequence to a gene in PDB or in other resources, but actually see if it's homologous in structure, and when there is variation, where is the variation? This is the lovely presentation that Shuchi showed, is how you can see the sequence variation and where it lands in the structure using that 1D, 3D tool. So all of this pieces together to give you a really rich readout that's so much farther beyond, the E-value is good enough, so let's just propagate the function. And that's the thing, like, that's why we're integrating KBase and PDB. That's the thing we want to empower people to do. So you want to, and of course you can apply the import apps to look at structure coverage of a genome. And I'll show you how to do that in a second. Identify gaps in the structure coverage, which is important as well. I'm going to try to drag this. Test for existing and completeness of protein complexes. I'll show you how to do that. And then import PDB and UniProt data into the annotation of the genome. So let's before I get to the future plans, because there's a lot still to do, I'm just going to jump out to the narratives and show some of these things quickly, because not all of it's totally obvious from what we've presented so far. So first of all, when you run this app, the PDB import PDB metadata in the KBase genome app, as you recall, this is the broad but shallow. So this queries every gene in your genome with against PDB and gives you all the hits. It doesn't just bring up this nifty table, which lets you search for the hits that you're interested in, and also link out. So for example, linking out to the PDB hit for any gene in your genome is wonderful, because now you get that structure context, and you remember you tie in through this link in the PDB, you're instantly tying the gene in your genome into all of that stuff that Shuchi and Brenda and Dennis showed, like wonderful tools to look at sequence alignment, structural homology, see how sequence variation relates to structure. All of that is at your fingertips, because you now have this link for every gene in your genome that has any homology against PDB. So that's the whole idea of this app, is put all of that, make all of that easy. And like something we'll be thinking about for the future is, for example, can we inject the sequence of this gene into that sequence alignment that Shuchi was showing, so that you would actually see your gene show up in that as one of the sequences in that alignment. So literally you could go to this structure page and instantly see where the variation of your gene lands on this structure. So that's something we're going to be thinking about for the future. I think it's particularly possible now that you have tools like AlphaFold that can rapidly make structures. And we're going to be looking at putting some variation of AlphaFold into KBase itself. So I think there's definitely there are many of these tools emerging now, and some variation that we should be able to put in. And just a reminder, this shows the method, all of the strands, the identity, and the e-values. This can help you craft your other query, like with the deep query tool. You can decide, oh, I want a lower identity than this, I want a lower e-value than this, I want to get a wider set of hits. And of course, the taxonomy, and this tells you whether or not RCSB and UniProt agree about the EC number, and that is also useful information. And of course, the hit to UniProt. And the papers themselves, as Jonica showed, can be quite valuable, because the title of the paper can be the function of the gene you're looking at. But to get it structural, that one thing I want to emphasize, we don't just put the data in this nifty table. We also inject the data into the KBase genome object itself. So the data is not just there for the user to see, it's injected programmatically into the KBase data model in the form of these ontology terms. So these are the EC numbers that were added. And what this is saying is that this is the EC number, and it's saying this EC number was because this gene is a hit for this structure in UniProt. And this is for UniProt. This EC number is in here because this gene is a hit in PDB. So it shows you where the EC number came from. It also sticks the co-crystallization information in here in the form of the model seed compound IDs. And as QZ showed, if you click on these compound IDs, it takes you to the model seed site with a picture of the structure and all the compound information and any reactions that compound is involved in. So you've got those links, not just in this table, but also in these ontology terms, and you've got the RCSB IDs themselves. One of the nice things the RCSBs give you is if you search in the genome for the letters RCSB, it will filter out to only the genes in your genome that have a hit in RCSB. So this gives you a very quick readout on the structural coverage of your genome. So this gives you 1,730 genes in your genome out of 14,000, but this includes all look for the word gene, out of 4,700. So roughly a third of this genome has a hit in PDB. And so for Arabidopsis, you can do the same thing. I think I have that. Yeah, here. So it's 8535 out of about 14,000. So I actually, interestingly, for the plant, over half the genome has a hit in PDB. Interesting with the coverage on the genes for the structural information. So I'm just, as cleanup batter, I've got notes on things I wanted to hit. The other interesting thing is complex completion. Because we have the structure information stashed in the genome, I plan on like connecting this structure information to the modeling, because the metabolic models have definitions of the gene complexes that form the enzymes that make each reaction happen. So we can compare those complexes to the complex information from PDB. And you can see this kind of exemplified perfectly if you look up something like ATP synthase. ATP synthase is a classic example of a gene that has a ton of subunits and the complex. And so by querying for this in this PDB query tool, we can ask questions like, does my luteus genome, is this complex complete here? So if we're going to annotate a complex to a gene in a genome, you know, one question to ask yourself is like, do I have evidence that this complex is actually completely represented in my genome? So for this case, you can see like one gene hits the first 3 chains, another gene hits the next 3, and another gene hits the final chain. So yes, this is and they're all homologous, they're all next to one another on a chromosome. So it's just a really nice, you know, demonstration of you know, how complex information can be another source of you know, validation for annotation, but also evaluating like whether or not, you know, my genome itself, is my genome complete, are the complexes complete? And again, this can be done like manually using the views that we offer you now and querying in clever ways. But I'm working on one of the next steps is to figure out how to do this programmatically, so that you can just with the click of a button instantly get a readout on how many of the complexes in your genome are complete versus appear to have missing steps. So it's sort of a complex-based gap filling that we'll be able to power in the future with this integration with PDB. On a similar note, the components is another interesting thing, because you see here, like this is ATP synthase, and of course you're seeing components you would expect to see. You're seeing phosphate, you're seeing ADP. Obviously, these are things that you know, these are substrates to this reaction. You expect to see them in the co-crystallization, but you're also seeing coenzyme type ligands here. So like magnesium, pyridoxal phosphate is everywhere. And that this doesn't mean it's a substrate. This is, in this case, these are PLP enzymes that just rely on pyridoxal phosphate as a coenzyme. So the co-crystallization information that comes in from PDB and is embedded into our annotations now here is actually providing you insights about the needs of these enzymes. And that's useful again for modeling, because if you knock out PLP biosynthesis or knock it down, you're not just going to impact, you know, an enzyme that uses PLP as a substrate directly. You're going to impact any enzyme that relies on this compound as a coenzyme. So having this rich information instantly at your fingertips, annotated into these genomes, and not just reference genomes, which we could just pre-compute all of that. But one of the big things about KBase is we're not just a reference database. We're working with your completely novel data that nobody's seen before. And so being able to rapidly and instantly query, fill these genomes out with this kinds of information is incredibly valuable. So it's interesting at the surface, but there's a lot of things under the surface. And I'm thinking about, you know, for the third-party developers among you, know that you have this data at your fingertips to do stuff with. You can, you know, tons of integrations that are powered by having this data available. I think that's the last thing I wanted to mention is the punchline of the Arabidopsis story. So the punchline is what you see from the study that Brenda did with the alignment. is that the two proteins in Arabidopsis actually align to two different parts of the E. coli gene that does with the RIBD function. And so one of the reasons that this function was mysterious for so long was that the most obvious gene that was the best homology hit, I think, which was 47390, you could stick it in E. coli and it would not do RIBD on its own. It did not fulfill the function. You actually had to put both genes in. So both genes are required. And now with that knowledge known in our current annotation of Arabidopsis, we now have these annotated. Both of these genes are annotated with this function. But the structure alignment actually shows you this really nicely. If you align both Arabidopsis structures to that E. coli gene, you get different parts of the structures aligning. And you get a whole other domain, which is a cog that still has an unknown function today in E. coli, but clearly has something to do with riboflavin biosynthesis. So there's a mystery for you that plant proteins are interesting because they can provide insights back into microbial functions because of the degree of fusion of functional domains and that happens within plant proteins. So yet another application here, and another value of having the Arabidopsis structures now available in PDB, as they've done, as Stephen and the team have done. That now that those plant structures are there from AlphaFold, you have that resource to go back and do those alignments to microbial genes and learn new functionality information, not just about plant genes, but about microbes as well. I think that hits. The only other thing I was going to mention was, if we go to QZ's narrative, see if I can find the right app here. Yes. So I'm going back here, and it's true that there's not a lot you can do at the moment with the structures and KBase. I think that will change soon. One of the things we can add, for example, is the structure homology search to PDB. They already have that functionality, so we can just dial into that through the APIs. So I think this will be a functionality that grows. But another point I'd make here is a value of putting your structures into KBase using this app is that it loads the structures, it connects it directly to your genomic information. That's why we ask for all the gene ID stuff. That's why we ask for this. It's connected explicitly to your genomic information, which is also here in your narrative. And so what I'd encourage people to do is put your structures in KBase so it's shared when you go to publish your narrative with your paper. It becomes a part of your publication, and anybody who wants to get that structure later can easily download it from KBase, just using the download controls here. And then when you do things like that structure alignment and PDB with the PDB alignment tools, screenshot the results of that and stick that in KBase in a markdown cell, much as we've done with this beautiful, RCSB logo. Actually screenshot that into a markdown cell in KBase so they can see that result as well. And now you're telling your entire scientific story in a narrative format that can sometimes be easier to digest than a journal, which is often highly filtered and cut down. Having the full logic laid out someplace, that's a big part of what KBase is about and what this interface is built for. With that, I'm going to go back to my slides. Sorry for the stream of consciousness here. So future plans for the KBase PDB apps. So Stephen is going to talk about some general future plans as well. I'm thinking specifically about the future of these KBase apps. So we currently, the Molstar viewer in KBase is currently only available on upload and specific upload of computational structures. But we're adding a widget, so that'll be available anytime. So remember, the structures get stashed as the structure, protein structures data type, and these narratives, like these protein structure data types, they're stashed in there. And so we're just going to make it so that if you click on this data type, the Molstar view would come up. At the moment, the viewer is very minimal. But we'll make it so that Molstar view actually comes up with your structures. So that's something we didn't quite get to in time for the workshop. We want to enable painting of other data in KBase onto structural views like sequence alignments and variation, much like what Shuchi showed. Probably less of a priority because I think the bigger priority is making sure that this kind of data in KBase can just be exported to the beautiful views they already have in the PDB site. So, however we end up doing it, that's something that's on our mind. Let's get all that data. Make it so that you can easily visualize all that data without too much pain. And figuring out how to do it manually. And then integrating more, just generally integrating more Molstar functionality into KBase. And you saw all the beautiful things that Molstar can do in Shuchi's presentation. The PDB query app, we want to enable query, like currently you can only paste in sequences. We want to enable query by feature IDs in KBase. Also querying compound structures by model seed or IDs or PubChem IDs, for example, instead of just NSheet. Saving query output as a sequence set so that you can connect it to other things. You can connect that query output to, for example, the structure import app. And then enabling query by structure alignment, which is, again, I mentioned that already. And then for structure import, like expanding the import to include the computational structures in PDB and other databases. So currently this tool only supports the experimental structures. And I would generally say this query app and the genome annotation app, that only queries experimental structures. And probably always, the query apps and the genome annotation probably always will just be focused on experimental structures, because to me, that's the valuable information we want to map onto the genomics data in KBase. And then for docking, we do have an AutoDoc Mina app in KBase, but it's linked to our older structured data type. So we'll be updating that to link to the new data type and enable you to save and look at docked poses later in tools like Molstar. More broadly, we want to link to modeling, so automatically compare model complexes with the PDB data, compare model reaction substrates with co-crystallized ligands, and be able to annotate models with coenzyme data, which if you're a modeler, like people are increasingly using this coenzyme data in metabolic modeling to better capture the behavior of these enzymes. So but when people do this, it's literally a year of manual annotation effort to get this data in. So having a tool that would automatically import this data from PDB would be enormously valuable. Structure alignment, we want to get this, examples of tools that are available in PDB or the Oak Ridge National Lab team is adding some tools along this line. And then experimental and computational structures, automatically linking these things so that you can compare your AlphaFold structures quickly to the experimental structures in PDB. As Stephen said, the computational structures really don't stand alone. You need the PDB, you need the experimental structures too, to make the best use, like pulling in the co-crystallization and really truth checking your folds. And then adding one or more computational structure pipelines. And we'd love to have any other ideas, and I'll acknowledge our many collaborators and the PDB and the KBase team, and of course, all the people who contributed to our workshop at the beginning of this project 2 years ago. And I will stop there